Hello, good afternoon. Um, sure to everything. Good afternoon. I am Lauren McGlone, and I'm going to do a unknown case study on my pathogen that I recently was given. Let's first start off by explaining what the case is and go through my methods of figuring out what I did and why and the reasonings of what I did. And here we go. So our case study, a 13-year-old female patient presents at her primary care practitioner suffering from a UTI, urinary tract infection, in which that have progressed to the bladder or kidneys. A sample of the midstream urine reveals bacteria in the urine. Alrighty, let's continue. So I got my sample on number 4B in lab, which is what the past slide was talking about, as in the little a 13-year-old girl had a urinary tract infection at a primary care practitioner, and it progressed up to the bladders or kidneys. So when I received a sample from the micro lab, I noticed that the bottom was very sedimentary to the nutrient broth. So what I did first was I vortexed it down and made it more motile, made the organism more motile, and more of the nutrient broth looks a little dim. So when I first received it, I did an oxidase test to see if it possessed the enzyme oxidase, in which nothing happened. And so from based off the case study list, it couldn't be Nicera or pseudomonads, so because it was a clear negative. So I immediately went into the enterobacteria family, because most of the microorganisms are from the enterics. So after it, after I produced the gram stain, it was a if it was a gram negative or a gram positive organism. It showed up when I used it from the broth. It showed up that it was gram negative rods, and I also inoculated the organism in a nutrient agar with the quadrant streak method, so I can see if there is multiple bacteria or if there was none. So to make sure that I had a clear, accurate results for this. So that's what happened. So what I first did was a triple sugar iron test to make sure that it was positive for fermenting lactose, or if it was possible for fermenting lactose or glucose in the metabolism of the organism, and to see if it produced hydrogen sulfide or carbon dioxide production to narrow down my results of the enteric family list. And I did another gram stain to make sure that it was a gram negative rod and not a gram positive rod, so that I can have clear, more accurate, efficient results. In which, when I did the gram stain, it came out positive for gram negative rods. And as if you can see here, there's tiny, tiny rods in clusters. So there's a lot of this organism. So here was the results from the TSI. And so what I noticed when I got it is that it was lactose and glucose fermentating, which means it was A over A, and it had no hydrogen sulfide production, in which would turn the butt of the tube black, which did not happen. And there's a little CO2 production, but not significantly enough to say that was positive for CO2 production. So So after I got the results from this, I decided to narrow it down to the klebsiella species because they both of them were A over A in both, and um and they did not produce CO2 or did any had no hydrogen sulfide production. So on the left, this was the broth, and on the right, this was my pure culture that I confirmed that there was not any other bacteria. So after I did that, I decided to do a urease test and a citrate test as well as McConkey agar to differentiate all the both the organisms down into one another. And I wanted to see if it would produce the enzyme urease and have the ability to utilize the carbon energy source. I also used McConkey agar since the organism was a gram-negative bacterium to differentiate their ability to ferment lactose and to produce a pH change in the agar itself. And I also used the SIM test to see if the organism modal or produces sulfur, which it wouldn't because the TSI would have showed hydrogen sulfide as production, and I wanted to see if it would produce indole. So after I had my results came back negative, the urease came back positive around in the middle of where I used the technique, a lazy S technique, and it caused a pH change and produced ammonia. But it was also negative because it was weird because around the circle, around the agar is orange, and that was unique to me. I also I also did the McConkey, which it looked really weak when I got it after incubating it for a little bit. It had a little of pink, but not much. It was very weak and very hard to tell, and so I decided to deem it positive. Also had a citrate test and also used the lazy S technique, and it showed that the middle turned positive, but around turned negative. So I just assumed that it was negative as well, and in which and then I did a sim I results from the sim test. It it seemed to be more modal from the stab site because it it went around the stab point, and I used Kovacs reagent to see if it would produce indole, in which it did not produce indole, and then it was clear positive that it was clear negative that there was no hydrogen sulfide production. And after getting all these results, I came to the conclusion that it was enterobacteria aerogenes instead of klebsiella pneumoniae, in which pneumoniae would have had more of a pink inoculation of pink colonies in the McConkey agar, and it would make a real strong positive, as well as real strong positive from urease and citrate. So conclusion, I was confirmed that it's an enterobacter aerogenes. Here's an electron scanning graph to show you the cellular arrangement of the Klebsiella aerogenes or enterobacteria aerogenes and what they look like under electron microscope. I thought that was pretty cool, so I just added it to my slide. So a little bit about enterobacteria aerogenes is usually found in hospital, in which it could cause nosocomial infections from daily sterilization techniques of the hospital, and it's usually a little bit more powerful, a more powerful infection since it's been used to sterilization and other things other things used at hospitals to clean. Is about 1 to 3 microns in length, which is about average for bacteria. and it is a petricious film, in which it, it makes the organism look like this, has things around. So it was highly modal. Is anaerobic facultative, in which it does not need oxygen. It needs oxygen, or does it need oxygen to survive? And individuals it usually doesn't cause disease in healthy individuals since it's found in the GI tract um, of your lumbar region area. So yeah, um, prevention of prevention and treatment of Klebsiella aerogenes is most susceptible to broad spectrum antibiotics, and if it is a bad infection, they usually use tetracycline or rifamycin or sulfadimidine, and usually tetracycline usually inhibits the protein synthesis of the bacteria, so it cannot produce proteins anymore, and and slows it down quite a bit, and so in which your body can produce immune response to get rid of the infection quickly. Um, and the prevention method that you could use is sterilizing catheters in hospitals, or simply just washing hands before doing any medical practice, and that will help significantly lower patients' chances of getting this this disease and or this bacteria in a place that definitely doesn't usually usually is found, and you know prevent infections that are not usually supposed to be there. And this will be the end of my presentation for Klebsiella aerogenes. I hope you enjoyed it, and you have a you have a wonderful day or night, or whatever time you're seeing this. Thank you, and I'll see you next time.